So today is another session of our monthly AORTIC North American series, part 5. This is a time we set aside in North America where we showcase our strength and the events that we have been doing on the continent. This is the hour with which we show collaboration, partnership amongst ourselves, and we want to know what others are doing, what we are doing, and pretty much everything is similar that we are doing on the continent and in the world. Again, today is also a World Cancer Day. And we in the international space have been working on in this field. A lot of things actually brings us together in different parts of the world that we are. Congratulations to us. And we are going to keep on working hard in this space. And hopefully in our lifetime, the outcomes of our patients, especially in Africa, on the African continent, will not be directly related to their geographical location. I know that in my lifetime, patients with any kind of cancer would have almost close outcome with the patients that I treat here in Chicago. That being said, another announcement I want to I'm pleased to announce today or share today is about the best of ASCO in Africa. That is going to be happening in person in Ethiopia. The date is June 27 and 28. So we will be sharing a lot of things. A lot of things will be happening in 2025. So today we have the celebrants in the room, the celebrant institution, which is Johns Hopkins University School of Medicine, who are going to be talking to us about their research work. So we have Professor Clayton Yates, who he is well known for his research in prostate and breast cancer in West Africa. He's going to be talking about his research work today. He's a professor of pathology, oncology, and urologic oncology. He's a director for translational health disparity and global health equity research. He co-leads the cancer genetics and epigenetics, and he is internationally recognized and an expert in prostate cancer health disparity research. And he is well known and most notable for his work centered on identifying multiple biomarkers for the prediction of aggressive cancers in African American patients with prostate, breast, and pancreatic cancer, which has led to the development of novel therapies for breast, prostate, and pancreatic cancer. Pretty much, he is what we call the bench to bedside and back. And in addition to him, we'll have Dr. Tison Manners, who is also a faculty in Johns Hopkins, and she is a past Fulbright U.S. scholar to Ethiopia. And I know that Professor Clayton is going to be introducing her more later in the talk, but she will be talking to us about omics-based analysis of prostate cancer in men of African ancestry. I'm always excited and I always look forward to this series because then I get to learn a lot of things that people are doing on the continent. And I know that in our lifetime, like I said, most likely the answer to cancer research will be coming from the African continent. Thank you again, Professor Clayton Yates, for coming on this hour to sharing your knowledge with us. Thank you.
Absolutely. Thank you for that wonderful introduction. And again, I didn't know it, but I was happy to learn that it's World Cancer Day. And I'm very honored that we're going to share some of our work from Sion and I that we think is impacting the world. I just want to say I partnered with AORTIC for many years, and I've been working in West Africa now. I had to think back. It's been so long. It feels like almost natural or home, but I think it's been almost over 10 years now. So, very, very, very excited to share some of our work and also celebrate a number of I think not only discoveries that my lab at Hopkins, and obviously I think most people know I was at Tuskegee University for many years before I moved to Johns Hopkins, but the work that people are doing in Nigeria is just amazing, and our consortium has really taken off. And I'll have to say it's kind of leading itself in the initiatives that they want to pursue that are affecting their communities and the patients they serve. So I'm really excited about that. So we'll go into a little bit about our building of our partnerships. I tried to push my slides. It didn't work. Hold on. Let's try again. Does that work? Okay.
Yes.
All right. So I have a few disclosures there. So as we know, and again, everyone on this call is understanding is that regardless of incidents, and we do have some challenges of capturing the amount of cancer incidents in some areas, particularly in West Africa. But what we do know is that regardless where Black individuals' lives, whether it's prostate or breast cancers, there's worse outcomes, right? And understanding if there's possible genetic, environmental, or social factors that are associated with that is really important. Because again, as we sort of talked about, we think the key to solving all cancer, not just cancer in patients of African ancestry, could be through understanding that this biastra, the genetics and the gene interplay that continue to drive aggressive disease. And so as a result, I happy to co-lead a prostate cancer transatlantic consortium, particularly from be the site leader for the U.S. But again, many of you know Falaki Odedna founded this consortium. Yeah, even when I was a junior faculty member and just being a part of sort of the day-to-day activities, and now along with Salah Rotimi, I call him my partner in crime, just leading efforts to empower institutions and or individuals to address some of their most pressing questions and concerns, particularly around prostate cancer research, as well as other cancers that we've evolved to over the years. Just a little bit about our consortium. I think we've grown, right? We were largely initially based in Nigeria, and I can sort of remember the first time I visited. I visited Federal Medical Center in Abuja, and I remember just meeting them. And again, it was at an aortic meeting when I first addressed one of the satellite meetings, addressed you know the community, and particularly the research community and medical community, and basically saw the need. And I can tell you that I promised in that meeting, that's why I spoke about my longevity, that I would not leave. I would always keep coming back, and I've been coming back for many years. During that time when we were there in Abuja, I traveled down to Lagos and some of the surrounding areas there. And you can now see where this CAPTC consortium started to grow. So we were largely in, sort of the Lagos, sort of South Africa, well, South Nigeria, excuse me, sort of area. And then now we've grown to all of these different sites across Nigeria, and now even into Cameroon, which we're really excited about. We just finished up, Dr. Solomon Rotimi finished up PSA testing and sort of awareness campaigns in Cameroon. And so we're getting that going with our colleagues there. And then now we're sort of also in other parts of West Africa, Rio, Ghana, as well as the Ivory Coast and Liberia and Sierra Leone. I think what is also important is that you sort of see this infrastructure building, right? So you see this CAPTC freezer on. Again, the first freezer, I remember we've been able to fund this out of a DOD grant that I got, was in Obekwata, right? So we right outside of Lagos, headed toward Covenant University, well, past Covenant University. And we were able to establish a freezer and a centrifuge. So we started collecting samples, and now we've grown into this multi-site consortium, which we're really, really very, very proud of. We started with a number of epidemiological and social behavior research. Obviously, Falaki Odedna is leading that, and Ernest Kananji, who is again a founding member or one of the early members of CAPTC, specifically in his training, and now has led, because he's from Cameroon, so he's now led this sort of expansion into Cameroon. So you can see how this sort of the turnover of leadership and people being involved and being able to take the resources within CAPTC and then bring those into his areas. And we're really excited about that and the studies that have sort of developed as a result of that, right? So we start to see difference in detection practice. We have risk factors associated with familiar cohorts, and even investigations of, if there's really environmental or genetic, and what happens in men of West African ancestry, particularly Nigerians who sort of travel to the US. Do we start to see the same profiles and so forth as well? And so all of these sort of behavioral studies, and the intervention of these behavioral studies with genomics is ongoing within CAPTC, and we're really proud about that. And we actually just received our second funding of heroic funding from the DoD to sort of continue those now into clinical trials. And I'll share with you one later this afternoon. So there's no secret that my particular areas have been around genomics, right? And again, the sort of impetus for me, as an African American was, if we really thought there were genetic differences, and we all understand the rates of disparities in the U.S., there had to be some ancestral sort of component to this. And just sort of digging into the data, we kind of don't have enough data to really make those determinations. So again, with an infrastructure like the CAPTC, we think it's really important to sort of add to this lack of genomic the lack of diversity in genomic data that's available, and sort of be able to understand that, and not only be able to understand what that diversity is, but make decisions that can now affect patient care, and as well. So, just sort of a highlight, these are some of the manuscripts that have the global impact of CAPTC investigators. I will like to highlight this African American, this Baswan A paper. We just were notified actually last week that it's won the Cancer Research Communications Distinguished Paper of the Year award. We will be getting presented with that award at ACR. It has our West African colleagues, Solomon Rotimi, Isaac Adelani, as well as our U.S. colleagues, who all came together to understand what is the relationship to African ancestry and vitamin D exposure. And now that is being recognized at the annual AACR meeting and the journal's most highly cited and publication for last year. So we're really excited about that. But we've also made other discoveries by sequencing the first prostate cancer genome for Nigerian men, and I'll show you some of that data as well today. So, when we sort of started this, as many of you know, we had to think about what would it take. So again, conducting omic data is very expensive, right? These samples have to be prepared in a certain way that they're preserved. This time intensive, funding intensive. So we did develop a processing protocol, and these are two publications that sort of ensue for that, where we develop a workflow from our pathologist, or actually from the surgeon to the surgery room, the theater room, to the pathology labs, understanding what it would take to properly fix those in a timely fashion, and now be able to, aliquot those samples so that they can actually be participating in research. We're really excited about that, and these are sort of two papers that, if you're very interested in that we're conducting these studies. And I'm also proud to say, in the next version of the TCGA, or the proteomics version of the TCGA, you will have Nigerian patients represented from our consortium that will be publicly available for everyone to analyze and to annotate and use for their analyses. And so again, we think this is real impact, and how our consortium has progressed over the years. I also want us to highlight what ensues when you do this kind of work, sir. So we have Solomon Rotimi. He's our partner in prime and co-PI within the CAPTC. And so when you start to be able to collect samples and ask questions that are germane to your community that are important, you get results like this, right? So we know, inflammation is really a big driver of disease, and how that affects prostate cancer progression and its relationship with testosterone. And so what Rotimi was able to show was that it's kind of different, right? So if you look at cortisol levels, which are elevated, and this relationship to testosterone, there are sort of this co-positive influence, and that the higher levels of testosterone correlate with the higher levels of inflammation. And we know they're both drivers of disease, particularly in patients of African ancestry. And so again, I just really want to highlight the collaborative nature and the empowerment that we're able to do by having these collective people at the table, and be able to utilize the resources in a way that is meaningful for the communities within the surrounding institutions. I also want to highlight, a really big project. And, I mentioned earlier that we sequenced the first prostate cancer genome from Nigerian men. And, I really wanted to highlight the gentleman here on the side, right? So these are our pathologists that sort of participated, in this development of our workflow, if you will, for our sample collection. And again, everyone had to come to the table. We met several times. We had pathology working groups to streamline the sample, the fixation techniques, so that we were all sort of coordinated, that we can conduct these sort of large studies. And what we found was that when you sequence men of African ancestry, particularly Nigeria, what we did was we actually had a hard time analyzing the data, because what we found was that majority of the variants that we were trying to call didn't align to the reference genome. So what we got was, in this sort of blue bottle here, is that these high level of variants of unknown significance. We saw that sort of in patients of African ancestry, African Americans as well. And then, but we were able to find some really unique findings as well, which there's a novel BRCA variant. So this RS799917 is unique to Nigerian men. We see that in Nigerian men and of African American men with high ancestry. All right, we only see that BRCA variant. And if you look at it, it actually turns out to be germline. So we see almost 100% of Nigerian men. We just followed up with a paper we published last year that in a larger cohort, it kind of came down to 92%, so 100%, but it's a really high germline variant. And what was really interesting, if you look at African American men, and so what we did was that we ran in genomic ancestry on each of these patients. If you sort of titrate ancestry, and what we mean is that the high African ancestry individuals are to the left, and the sort of more admixed population patients are to the right, you sort of can titrate out the variant. So that 68% is representing 6% of African Americans, but African Americans with high West African ancestry have more closer to that 90% that we see in Native African men. So again, we think this was very important, and this was something that that is being followed up on, particularly in Nigeria, where again Solomon Rotimi and the CAP-TC now has partnerships with AstraZeneca, so we can understand what this means to current therapies such as PARP inhibitors and et cetera, because we know that these are biomarkers that allow people to be enrolled in these type of therapies. We also were able to look at somatic mutations, where we not only saw BRCA1, we saw entire multiple markers, right? Largely around the DNA damage repair genes, right? So if you look at the BRCA genes, APC, and even a sort of a newer gene within the DNA damage repair pathway called SPOP. And, collectively, we see that these mutations seem to be associated with patients of African ancestry. They were there in Native African men. They're also there in African American men with high African ancestry. And we think this is really important as we're starting to understand the genomic landscape of patients, particularly as it relates to targeted therapy. And if you look at what these DNA damage repair genes sort of influence, it influences the things that we know are important in tumor genesis, tumor immunity, right? Adherence junctions, right? How's signaling pathways occur. And particularly in this immune sort of pathway, we thought was really interesting, because prostate tumors have largely, not done well with immune therapy because they've been immunologically cold tumors. But here we are seeing that, this sort of tracking in these African American or African ancestry patients, that we sort of see this sort of increase in sort of this immune response, particularly at the genomic level. So in order for us to sort of dive into that, we wanted to go even further. So we wanted to understand what was the transcriptome profile impact of these genomic signatures. So we took one of these particular genomic markers called SPOP, and this is what you see over here on the left. And we actually overlaid these SPOP mutant patients with their gene expression. That means this is their transcriptome. And what you can see is that even within a mutant population, patients with 50 to 80% ancestry have differential gene expression. With patients with over African ancestry, again, all with the same mutational profile, have a differential gene expression. And so it's not just a mutation impact on a gene, it's ancestry-based mutational interrelationships that seem to be influencing gene expression. So if we start to look at what this looks like in our African American and African ancestry individuals, you sort of see this SPOP increased. You sort of see that also again in correlated with our patient populations. What are genes that are associated with this? Inflammatory type genes, right? So these inflammatory markers, interferon gamma response, and this interferon signature, and we don't see that in patients who don't have this sort of mutational profile. And if we do spatial profiling on these individuals, and we have African Americans, and now we're sort of adding a West African cohort to as well, we sort of see again what we thought were immunologically cold tumors. They seem to be infiltrating now. These immune cells are present and or infiltrating in the tumor parenchyma in our African American patients. So we see high PD-L1 and T cell infiltration, and we also see a CD8 163 macrophage sort of infiltration that is occurring in our sort of mutant profile patients. And not only see that at the transcriptome level, we see it at the proteome level as well. So what does all this mean? So we're not the only people that have shown this. There's a manuscript in Nature, came out from Vanessa Hayes' group in South African individuals, and back-to-back publications that show very nicely that, men of African ancestry, right, particularly West African ancestry, have this sort of DNA damage repair genes, and this seems to be driving tumorigenesis. Now, what does this sort of do, right? So if you look at, particularly in this case of SPOP, which is again one of the newer DNA damage repair genes, its role is an E3 ligase, and its job is to degrade these intercellular proteins, PDL1, AR. But you can imagine when it's mutated, it's not doing that. And so we think this is a reason why we see this upregulation of PDL1, upregulation of androgen receptor. And so it's really interesting because a colleague of mine, Emmanuel Integrarakis, did a phenomenal clinical trial. So if you look at SPOP mutant patients, again, these patients should have upregulation of PD-L1 and androgen receptor because the SPOP is mutated. And if you look at patients who progress to therapy, SPOP mutant patients, we sort of theorize, or at least Emmanuel theorized in his manuscript, that they should never be triaged to chemotherapy. They should stay on androgen therapy even if they fail frontline androgen deprivation. And so if you look at the survival outcomes, he's absolutely right. So the patients who have SPOT mutations, who failed frontline AR therapy and got second line AR therapy instead of chemotherapy, which is the standard of care, they have overwhelmingly survival benefit compared to, again, patients who got African ancestry. So again, this is why we do this work. It's really important that we take this and try to translate this into clinical practice, or at least into a clinical trial. And I'm really excited to say is that when we looked at our African American patients based on this study, we saw the same case. So SPOT patients who have African ancestry have increased androgen receptor signaling, right? And if you look at this, partnering with cell-free DNA companies like Guardian, we not only see that these patients have increased androgen receptor signaling, that Black patients who have SPOT mutations actually respond better to androgen deprivation therapy compared to other patient profiles. And these patients have a 10-month survival advantage compared to wild type or even European men with SPOT mutations. So again, this is really important that we're taking basic science discovery and translating those and making a meaningful therapy or making a meaningful contributions and putting it into the standard of care so we can better understand how our patients respond to certain therapies. But we wanted to go even further. And I'm happy to announce that we are just been, funded for our first clinical trial in Nigeria, our first, drug-based clinical trial, where we're going to basically utilize a novel therapy developed at Johns Hopkins. It's called bipolar androgen deprivation therapy. And I'll explain it to you in a minute. But what this therapy does is that instead of doing androgen deprivation, as we know is the standard of care, when patients ultimately are going to progress to castrate resistance, those patients through super physiological androgen or doses of testosterone, we can actually return that back on. So we can turn that androgen receptor back on and they become responsive. So if you look at this sort of diagram on the left, it's like, look, androgen receptor we know is driving tumorigenesis. There's been a myriad of treatment modalities, orchiectomies, medical castration, and even biochemical castration, to sort of block this. But we ultimately know the patients relapse. And so our sort of approach is that actually if you oversaturate these patients, you can actually turn this back on. You can turn this androgen receptor back on. And so if you sort of see this castration sensitive, you go into this dormant phase. And as patients develop castration resistance, if you sort of flip, sort of cycle them over into an high dose androgen deprivation, at high dose androgen therapy, you can actually resensitize the androgen receptor, and we call that bipolar therapy. And so again, since we saw this DNA damage repair in our African Americans and patients with DNA damage repair genes in these clinical trials respond better, we opened up our first clinical trial called Superman, right? And so this is just a little, synopsis of the previous trials. Again, this is testosterone prostate cancer trial. The first trial was named Batman. The second trial was the Restore, and the last one is Transformer. But the overall, take home from this is that, cycling, patients over to superphysiological after castration resistance or confirmed metastatic castration resistant disease actually resensitizes those patients, and that the patients actually experience a better quality of life as well because of, we know the side effects of androgen deprivation. And so our trial, again, named Superman, is a single arm trial where we're basically doing a lead-in study. So patients will get standard of care and a deprivation, but after 6 months, we would then cycle them to high testosterone, and then we would cycle them to ADT, to BAT therapy to ADT, to hopefully prevent these patients from progressing to late stage or end stage disease. And again, this is going to be led by Dr. Professor Popola at University of Ilorin, and it will be conducted in Nigeria at University of Ilorin as the prime site. And again, we're really excited for that. And so just to sort of end, we've created these multi-omic cohorts that we think are important. We think these multi-omic cohorts are necessary to understand. So we have, not only this genomic data, we have transcriptomic data, we have methyl-seq data. We're creating these methyl sequencing and single cell sequencing annotations so that we can really dig into what the contribution of antigen ancestry is and how that sort of plays a role. Tiana's going to talk about this more in depth and some more concrete studies in a second. But just really quickly, we've been able to show that from the transcriptome level, Native African American men have different set of signatures than African American men as well. And if we sort of look at what that is, there's the differences in inflammatory scores as well as immune signatures. And then we see that the immune cells that sort of infiltrate in this as they're all surrounded around these myeloid. So these myeloid suppressive immune signatures appear to be driving the disease. And if we can start to capture these in AI models, so we're utilizing these novel AI three-dimensional models, which are developed here, that we can sort of capture these immune cells by utilizing machine learning approaches, which is here, physiologically annotated. We can start to create these three-dimensional models where we can capture this without that fancy omics. We'll be able to capture immune cell infiltrations, particularly from H&E, by utilizing, again, these machine learning AI approaches. And we're already able to do that, and they're able to show that African Americans, particularly Native Africans, have this more immune inflammatory signature. And a really talented postdoc in my lab was able to show that this inflammation is not only within the tumor parenchyma, but it's also in benign ducts as well. And we're able to capture this again through AI-based machine learning, identifying these immune cell types. So just to sort of close this part of the story, we think this is important. Personalized medicine should be for everyone, and that we think this in-depth biological approach is essential for us understanding that. And then hopefully we feel this time. You see our clinical trial the design. This is another one called Supple-Cel-T, which is a immune therapy where all of a sort over-enrolled African American patients, and it's a sort of immune therapy after castration resistance, very similar to our Superman trial. And he saw 2 times better response in our African ancestry versus European ancestry. So we think that's really important. So just to sort of talk about, again, capacity building is important. These are the people who actually train in my lab. They now have their own labs and they're conducting the research. This is a picture of our consortium. This is my lab who did all the work, and the individuals from our team that are sort of on the ground, and really proud of that. And I'm happy to announce that we're going to take these small studies and now launch it into what we're calling Standby, which is a 40-patient recruitment multi omic approach, where we're starting to understand what the intersection with these and environments, and not only in prostate cancer, but breast cancer patients and pancreatic patients as well. And I'll stop there and turn it over to Sia. Should we wait for questions at the end?
I think we should do, because I think you will have a lot of questions. So probably we should move on to the next speaker, and then we'll do questions at the end.
Okay. All right. I'm really proud to introduce Sion Minas, who is an instructor at Johns Hopkins. Sion did her graduate work at Georgetown University and her postdoctoral fellowship at NCI. She has been celebrated. She is an AACR scholar. She is a full box scholar. She has published many papers. You'll show all the wonderful work she has here. And I think what she represents for Johns Hopkins is a new initiative there's no doubt that we've been a leader in global health, but bringing Sion in and her work, particularly in African American men with prostate cancer in West Africa, she's now taking that to Ethiopia. So she's a resident in Ethiopia and is part of our global sort of oncology approach, where we're actually going to have boots on the ground and be able to take some of the strategies that we're finding out from this sort of omic data and really implement that in the community. So I'm really happy to introduce Sion and hear the work that she's going to share with you guys today.
Thank you so much, Dr. Clayton, for the introduction. You guys can hear me, right? Yes, okay, great. And also for AORTIC for this invitation to present my work. Today I'll be talking about how we used omics-based approach to study prostate cancer, particularly in men of African ancestry. Right. So as you all know, both in the US and globally, men of African ancestry have the highest prostate cancer burden. If we look at the mortality rates, turn on my pointer. If we look at the mortality rates in 2020, globally, we see that a number of African countries have the highest mortality rates. If we look at the CDC data between the year 2000 and 2020, we see that African American men continue to have the highest incidence and mortality rates. And of course, the gap still persists. So to really investigate the cause for prostate cancer disparity, we employed a diverse study cohort involving African American men and European American men, a total of 2,000 men from the NCI Maryland prostate cancer study. We also utilized the prostate cancer transatlantic consortium that involved Nigerian men. Also, the NCI Ghana prostate cancer study that has about 1,000 men in this study. So what we have is basically from most of these men, we have urine, blood, and saliva sample, and of course for the cases we have tumor samples. So these biospecimens were used to generate this multi-layer omic data that we integrate to basically inform biomedical research, such as mechanistic studies to better understand the disease. Also, in laboratory studies that aim to find therapeutic targets. This type of approach could also guide prevention research and also improve clinical care, where it could lead to improved disease classification or leading to more better risk stratification or treatment stratification so that the right patients receive the right treatment, and also for discovery of biomarkers. So today I will demonstrate how we utilize this approach to really investigate if ancestry really plays a role in variation in host immunity, and also what does that variation in host immunity has a role in prostate cancer, also how the role of fatty acids in prostate cancer. So first I will talk about how we investigated if ancestral variation in host immunity exists, really, and if it does it contribute to the excessive prostate cancer burden we see in men of African ancestry? So we utilized the same study cohort I mentioned earlier. We analyzed 92 immune oncological proteins and over 3,000 blood samples from African men, African American and European American men for the study. And we basically, to investigate, to measure these proteins, we use what's called proximal extension assay by Olings proteomics. Basically, this assay has antibody pairs that are linked to these oligonucleotides that are complementary to each other. So only when these antibody pairs recognize and bind to epitopes that are in close proximity can the hybridization occur, leading to basically the detection of this protein. And this is a very specific assay, basically allowing us, which is why it allows us to measure 92 proteins simultaneously from just 2 microliter of blood. So to really investigate whether ancestry plays a role in the levels of these immune oncological proteins in blood circulation, we perform what's called unsupervised hierarchical clustering to really see, do we see clustering of these, or can we separate these population groups by just using these immune oncological proteins that we measure from blood samples? So interestingly, so by using this 82 out of the 92 that have passed QC, just using these 82 proteins, we were able to group Ghanaian men, which is indicated in light blue here, and the dark blue is African American men, and the light green is European American men. So we were able to actually group them just by the measurements of these 82 immune oncological proteins from the blood. So really showing that, yes, ancestry indeed could play a role in affecting the levels of these immune oncological proteins in blood circulation. And also, as you can see, Ghanaian men actually cluster closer to African American than European American. So to really further investigate the role of ancestry, what we did was basically we determined degree of West African ancestry using the DNA we extracted from samples from African American and European American men, and we were able to determine the degree of West African ancestry these men had. And we evaluated if these 82, that the proteins you see at the bottom, if these 82 immuno-oncological proteins can get influenced, their levels can get influenced by a degree of West African ancestry. And this is in men without prostate cancer. We didn't want to have that confound our findings. So we wanted to investigate this in just, quote unquote, healthy men. So what we saw was that, out of the 82 proteins, 39 of them that you can see, like where I put red stars here, 39 of the 82 were actually significantly influenced by a degree of West African ancestry, even after correcting for multiple testing. Particularly, some of these chemokines were highly correlated with degree of West African ancestry. Specifically, for example, CXL5, as you can see, 50% of the variability in the level of CXL5 in blood circulation could be explained by degree of West African ancestry that these men had. Similarly, 40% for CXL1. So we then compared the level of these proteins across the three population groups. So comparing the level with Ghanaian men, African American men, and European American men. So CXL5, CXL1, 11, and MCP2, the level of these chemokines were highly elevated in Ghanaian men, where they're the lowest in European American men. But for MCP1 and NCR1, the levels of these chemokines are elevated in European American men versus they're the lowest in Ghanaian men. So showing this African ancestry related trend. So we were trying to understand why we saw some of these chemokines to be highly elevated. And I found this review paper at the time where really showing that this receptor called DARK, which is Duffy antigen receptor for chemokines, basically what it does is it binds to chemokines that it targets and it sequesters them away from blood circulation. So it sort of serves as a sponge, sort of regulating the level of some of these chemokines that exist in the blood. But interestingly, this DARK is also what malaria parasite uses to infect red blood cells or erythrocytes. So basically, because of adaptation, and a number of individuals in sub-Saharan Africa have acquired this resistance to malaria infection, meaning that they lost this receptor, the DARK receptor on their erythrocytes. So you see a number of men or people of African ancestry, for that matter, are DARK null. So they lack this receptor on their erythrocyte. They could have it in the endothelial cells, but they lack it in the erythrocyte as a mechanism of having a resistance for malaria parasite infection. So what does this mean for the chemokines? These are the chemokines that have been found to be targets of DARK binding. And as you can see, there's some parallel where we already saw CXL1, CXL11, CXL5, and CXL2 as well to be CCL2 to be elevated in men of African ancestry, then these are the ones that are actually being targeted by DARK, meaning that loss of DARK on the erythrocyte in these men leads to those erythrocytes being unable to actually sequester them away, these chemokines from circulation and from regulating them, their levels in circulation. So to really see, and we are currently doing more study to really confirm if that's what's happening in our study cohorts. And we have genotype, the dark polymorphism. So we hopefully will have something out soon on that. And so to really see what's the functional implication of this sort of differences that we're seeing in the levels of these immune-oncological proteins, we group these immune-oncological proteins by biological processes such as apoptosis, autophagy, chemotaxis, promotion of tumor immunity, suppression of tumor immunity, and vasculature. And we tried to compare these scores by population groups. And basically what we saw was these immune-oncological proteins are associated with chemotaxis and suppression of tumor immunity were the ones that showed sort of ancestry-related trend, where their score was higher in men of African ancestry than European ancestry. And to see what the clinical implication of all of this is with these biological processes, to see which ones have the most impact on survival of these patients, we perform a survival analysis, and from our data, it seems like suppression of tumor immunity is the one that has the most impact on the survival of this patient. Basically, men that have the elevated level of suppression of tumor immunity are more likely to die from all causes, from prostate cancer, and also from any cancer. And that's even after adjusting for various factors. And we were also able to show that having high level of suppression of tumor immunity is associated with increased risk of being diagnosed with original or metastatic disease. And this is also, we were able to see similar trend, even though we didn't reach statistical significance, even though the association is less, still we were able to see that trend also exists in European American men. So basically, to conclude, we saw that the immunome of Ghanaian men resembled the immunome of African American men than European American men, and we did see this chemotaxis and suppression of tumor immunity to be elevated in men of African ancestry, and this could mean a lot of things, right? So this could mean that we may see different tumor microenvironment in men of African ancestry, and it could influence also metastasis formation, and the type of immune cells that are going to infiltrate could be different. So it could be a distinct tumor biology, basically, what we're investigating. And the fact that we saw elevated suppression of tumor immunity in the blood circulation, and the fact that that also associates with lethal prostate cancer, could mean that these men could be actually benefit from therapeutically from immunotherapeutic strategies that are tailored to target immune suppressive pathways. So whenever we are designing, especially immunotherapy related trials, we should make sure we have representation of men of African ancestry, because it seems like from our data, those may be the patients that would benefit the most from this approach. So I will also continue quickly to the next part, where we also try to use these measurements to see if we can come up with a predictor model to figure out which men at the time of diagnosis are more likely to be developing lethal prostate cancer, or which men are more likely to die from prostate cancer, because that's a major clinical challenge right now, because majority of the prostate cancer patients will not die from prostate cancer. They will die of other causes. So how do we differentiate these men so that they get the timely treatment, and those that may not die from it, they don't receive unnecessary aggressive treatment? So to do that, we employed AI to see if any of the proteins we measure could be predictors of which men will be dying from prostate cancer. So long story short, basically what we found was pleiotropin and TNFRSF9 were the ones out of the 82 proteins that we measure. They, along with NCCN risk score, which is what's currently being used in the clinic, were able to predict prostate cancer mortality with 90% accuracy using these two proteins, along with the NCCN risk score. So, and looking at this, how these proteins could stratify these patients, and I forgot to mention actually, so when we did the study, we were able to show that we couldn't find any of these proteins to be predictors of mortality for European American men. So this was only working, these proteins were only being predictor for mortality of African American men. So we went ahead and tried to see how these proteins were able to stratify these patients. So if men at the time of diagnosis had low levels of both of these two proteins, as you can see, even after 15 years of follow-up, majority of these patients survive. And similarly, if these men had low levels of either one of these two proteins, still majority of these patients will survive. But these men at the time of diagnosis had elevated level of these two proteins. We see that about 60% of these patients will die after 15 years of follow-up. And the hazard ratio for this is about close to 6 times. So these men are 6 times more likely to die of prostate cancer than the other subgroups of men. And this is just for African American men because these two proteins were not being predictors for European American men. So we didn't have survival information, mortality data for the African cohort, so we couldn't do this study. That would be something we need to follow up on. But we did measure these 82 proteins in Nigerian cohort as well. And we were able to compare cases and controls. And what we saw is, again, pleiotropin is coming as to be significantly elevated in cases than control, suggesting that maybe this protein could be relevant for this population group. So this is something we would have to follow up on. So to conclude, these two proteins along with NCCN could be used to predict mortality, prostate cancer mortality with 90% accuracy. Of course, this could be used for risk stratification at the time of diagnosis. So, and this is the population group that needs it the most because African Americans or men of African ancestry are the ones that are really bearing the prostate cancer burden. So, of course, this all needs to be validated before it could be used in the clinics. So, briefly, I'll briefly go through the fatty acid omics data we've done, trying to see its role in prostate cancer. Fatty acids play a significant role in resolution of tissue inflammation. There are these recently identified specialized pro-resolving mediators, small molecules that are derived from polyunsaturated fatty acids that play a key role in really ensuring that inflammation gets resolved so that it doesn't kind of proceed and become chronic. So we went ahead and measured 24 fatty acid in 3,000 men, including Ghanaian, African American, and European American men. And what we saw was that in Ghanaian men, we saw trans fatty acids and omega-6 fatty acids were lower compared to American men. Actually, we didn't see significant difference in the level of fatty acid in African American and European American men. More so geographical, I guess. So we saw omega-6 was lower and also trans fatty acid was lower in Ghanaian men than American men, whereas omega-3 was elevated higher in blood circulation in Ghanaian men than African American or European American men. So to see if these circulating fatty acids are associated with elevated prostate cancer risk, we went ahead and did volcano plots and compared their levels in cases versus control. Even after accounting for multiple testing, we saw that, as you can see, the red dots here in all of these three population groups. So this is Ghanaian, African American, and European American men. And as you can see, these three trans fatty acids, those are the different trans fatty acids. that are indicated in red dots here. So you saw them to be elevated in all of these three population groups in cases over controls. So basically, we saw that even when we were doing adjusted analysis, we saw a dose-dependent increase with increasing trans fatty acids. We saw increasing risk for prostate cancer, and that was the same across three population groups, really suggesting that this association may be real, actually. So obviously, it needs to be validated and prospective cohort, but suggesting that trans fatty acids could be a risk factor for prostate cancer. So basically, to conclude, we did see a significant positive association of circulating trans fatty acids with prostate cancer, and that was independent of ancestry or geographical location. That was all aboard. So to really summarize, I hope that I demonstrated how using this integrative omics-based approach to study cancer health disparities could really help us inform biomedical research, where we can now investigate the role of these two proteins in prostate cancer, also trans fatty acid in carcinogenesis or prostate carcinogenesis. So we can do mechanistic studies in the laboratory to really see if there is if these are causative relationships, and sort of promoting prostate cancer progression or even tumorigenesis. So it could also guide prevention research, where using this data, it suggests that maybe reducing trans fat intake may be a good idea, but obviously this could be further collaborated with prospective studies and investigating if this is true association or causal association. So this also could lead to improved clinical care, where we can use now these two proteins for risk stratification at the clinic, obviously after validation studies. So I think, yeah, obviously a lot of people contributed to this, including NCI and groups from University of Maryland, Ghana, of course, Nigeria, and also different Georgetown as well. So with that, thank you so much for your attention, and I'll take any questions.
Thank you so much for that wonderful presentation. And I see that there's already hands up. If you have questions, please raise your hand. This is a summary that just means that tumor does matter, and there's a lot to be said about that. So I see your hand raised by UCSF Global Cancer Program. Can you unmute yourself? Okay.
Hi, my name is Mac Roach, and I'm a radiation oncologist, and I've been involved in prostate cancer clinical trials involving African American men, and written many papers on prostate cancer in African American men. Is it possible for me to share a slide or two? I wanted to make a couple of points. Those presentations were excellent. But I think it's important for people to remember to distinguish the problem with incidence in prostate cancer in African American men versus outcome after treatment. Because it's very clear that prostate cancer is much more common in African American men, but the notion that the prognosis is worse based on data from prospective large phase 3 randomized trials, the evidence is that African American men do not have biologically more aggressive disease if African American men get the same treatment for the same stage of disease. So I don't know if I can share one slide I wanted to, and if somebody can let me share.
Okay. So while team members are working on that. Okay, yeah.
Okay, yeah, thank you. So this is from a review article that we published recently, and this is a summary at that time of 12 randomized trials in which people went back and did a post hoc analysis to look at, was there any evidence of a difference in outcome between Black men and white men treated on phase 3 randomized trials. And in summary, the only trial that found that African American men did worse is the one by Thompson, which is in the metastatic setting. There were 8 studies that found that race was not important at all. And there were 3 studies that showed that African American men did better than white men. And those 3 studies happened to be larger studies and more recent studies. And one of them was mentioned, the one involving sickle cell T, which is an immunotherapy. And in fact, I believe that prostate cancer is more common in African American men because of issues related to immunity. So we're in alignment with respect to some of the different biologic considerations that may be involved in the incidence of prostate cancer. But if you look at the results from randomized trials, the fact is that African American men do not do worse when you get the same treatment for prostate cancer. And we have a paper which is currently in press in JCO Informatics, specifically focusing on the use of artificial intelligence in more than 5,000 patients, in which we looked at, these are all patients treated on randomized trials. And in fact, what we show in our paper, again, in press, is that African American men do a little bit better. And in fact, in our title of the paper, we make the
really
point
quickly,
that
so from our perspective, like right now, we were focused on our African men, and there's really not a lot of folks. So we do have these studies in African American men, we absolutely, but the focus of this and our studies was to understand the biology, because what we see is solutions not being transferred to West Africa. And so
right.
if I misunderstood the take home of our conversation,
No.
So they want to go through, because you could give your own presentation very nicely, but at least we thought was the purpose of this aortic meeting was to talk about how do we take these solutions into West Africa, because these trials are not being conducted. We don't understand many of the standard of care options that are available in the U.S. are not available, so we have to sort of pare those down. And so there's a huge difference, and the biology makes a difference as to what medicines or treatment strategies you pair it with.
So that's exactly my point. So we're in total agreement. My point from the beginning is that the key, the gold, whatever, is to find out why prostate cancer is more common in Black men. So that, I think, is the single most important thing that we need to get an understanding for. We know that in the U.S., and we know that in the islands, in Jamaica and other places, and we know that in Africa. And that's what the research that you all are doing, I think that's where that research is particularly critical. And all I'm sharing with you is the fact that for someone who's been doing this for over 30 years, that the fact is that, yes, it's important for you all to bring the standard of care and treatment in Africa up to an appropriate level. But it's also important to understand that the key challenge is figuring out why prostate cancer is more common in Black men.
So thank you for that. I think at this point, the question is going to be, or maybe the invitation is going to be that for us to continue this discussion, probably UCSF, if you can talk about your data to the whole team, would like to see that data. And I just wonder, from the data that you showed, that how many Black patients were actually represented in those trials. We do know that most of these globally conducted trials, when they do that, they will tell you like when 5, 6, 7, 8 Black people were enrolled on the trials. And is this adequate enough? I mean, this is
we had 900.
Yes.
900, and in fact, on our trials, 25% of our patients are Black. Right. So I have another study with 2,592 patients treated on a large phase 3 randomized trial, and 20 to 25% of our patients are Black.
So I'll tell you, so Sion didn't present the data, but in our data from our American cohort with 3,000 patients, White Americans have worse outcome.
Which ones?
In our data, the one we just presented, in case of 3, we're writing it up now. African Americans have worse outcome. in
Yeah,
Maryland.
but those are not, see, those patients
So-
were not treated on randomized trials. So you
So
also
no,
have to
you-
look
so
at
yeah,
the quality of
we're
care.
not arguing that the trials is because if a man gets in a trial, but then we still have worse outcomes. So like you said, it's like you
No,
have
we
to
don't.
understand the
That was the point
biology.
of that table, is that we don't have worse outcomes if we're treated on phase 3 randomized trials. That's the point.
Okay.
In population-based studies, 30 years ago, everybody told me I was wrong. They said prostate cancer was inherently more aggressive in Black men. And I said, well, prove it. You can't use SEER data and you can't use other population-based data to compare outcomes. You need data from randomized trials where you can adjust for stage, grade, quality of care, and treatment. And that's what those 12 studies I just showed you were, all data from randomized trials.
Okay. So then I think the next question I think I can read from there is we were wondering about data from other parts. I mean, Africa is not a monolith, and the hope is that we can also see some work done in East Africa and North Africa, and we start discussing that. And then moving on into representation of Black patients in clinical trials, not only in prostate cancer. Right. And this is something that we've been talking about on the continent. This is something that we've been talking about in AORTIC. And we have a whole clinical trial special interest group that is actually dedicated in addressing that. So is there any other questions? Do we have any other questions on the team? So for those that
I'd
is
just like
trying.
to add to point that, and I think Francis Makoka is on as well. We're launching our East African cohort now. So we were in
Okay.
Kenya last month with Francis. Sion is based in Ethiopia every day. She's Ethiopian and based in Ethiopia. So we're starting to launch, similar. We don't have the clinical trials. We'd love to partner with the clinical trials, but from the basic omic research so that we can understand match therapies with certain treatment modalities, or excuse me, gene signatures with certain treatment modalities. We would love to partner with AORTIC for that, so that we identify signatures, we can triage those individuals to the appropriate care that they need or clinical trial. So we'd love to partner with you on that. But yeah, we're launching our 40,000 patient enrollment right now. And so we're really excited about that. And that is absolutely the goal. And it's across breast, prostate, and pancreatic, by the way, not just prostate.
Wonderful, wonderful. And hopefully you will be giving us updates in a couple of months or years, so that, this is going to be a continuous effort. So as we are rounding up this is top of the hour. Thank you for everybody that showed up. Thank you to our presenters, and for our contributors too. So this time next month, what institution is coming up next? We are going to share that on our website with you all. So thank you again, and I will be ending this, and God bless you. Okay, so we have, I think, Mayo Clinic. Mayo Clinic is going to be presenting next month. So please keep it a date with us. This is always an interesting presentation, always fascinating and mind-blowing. So next month, first Tuesday of every month is when we meet again. All right. Thank you very much, and bye-bye.